OMNI-RÉUNIS Super Spreader Seminar Series. And for everybody to be aware, we are recording the seminar. My name is Arne Rückert, and I'm an Associate Director of the OMNI-RÉUNIS Network. I'm also the Managing Director of the Globalization and Health Equity Research Unit at the University of Ottawa, which is located on traditional territory of the Algonquin Nation. And I briefly want to read to you the official land acknowledgement statement. We pay respect to the Algonquin people who are the traditional guardians of this land. We acknowledge their long-standing relationship with this territory, which remains unceded. We pay respect to all Indigenous people in this region from all nations across Canada who call Ottawa home. As one of the deadliest zoonoses known to us, rabies still kills around 60,000 people every year, representing both a heavy public health and economic burden. Yet it is 100% preventable if you address the disease through a One Health approach. Can one sector eliminate rabies on its own? Certainly not. The COVID-19 pandemic and its long-lasting consequences have reminded us that one sector alone cannot effectively tackle any zoonotic disease, knowing that animal, human, and environmental health are intrinsically interconnected. There's currently an uptick of rabies in Canada and in Ontario as well, which has led to a number of policy interventions and actions, including some highly contested ones. And I'm thinking here of the recent banning of the importation of dogs from more than 100 countries, which garnered quite a bit of media attention. In this context, One Health modeling can help us understand how current control methods will affect rabies spread, whether the projected and used control methods are effective, and as such can inform a cost-benefit analysis of One Health policy interventions. Our presenter today is Dr. Emily Acheson, who is a spatial epidemiologist at the Public Health Agency of Canada. She recently completed a postdoctoral fellowship with Dr. Patrick Layton at the University of Montreal using agent-based modeling to better understand the effects of control interventions on raccoon rabies in Southern Ontario. So without further ado, please welcome Dr. Emily Acheson. The floor is all yours, Emily.
Hi, everyone. Okay, I will share my screen now, okay, to make sure that this works again. I had it the first time, so it should go. There we go. Okay. Is everyone okay? Everybody can see my screen okay? I'll take the silence as a yes. Yeah. Okay, that's great. So thank you everybody for coming to my talk today. I'm really excited to share these results because this is actually the first time that I'm sharing our final results, which I just got in the last few days, and they're very interesting. So I'm very excited to share that. I would like to start by saying that this is a collaborative project with Patrick Layton at the University of Montreal, as was said in the introduction. Thank you, Arne. And so I've been doing my postdoc with him, and we're just wrapping up this project as I start working with FAC. And so we're doing this in collaboration with the Ministry of Natural Resources and Forestry. And this is because there has been an outbreak, which is ongoing in Southern Ontario with raccoon rabies. And so we were actually approached by the Ministry of Natural Resources and Forestry. This includes Tor Buchanan and Larissa Nytek, who are working with me on this, in order to use agent-based modeling to see how control interventions are working in Southern Ontario, to see how best we can optimize those controls and limit the spread and limit disease in this region. So just briefly, my slides are going to go here. There we go. There we go. So I'll just discuss very, very briefly, since there was already a great introduction about this, what rabies in Ontario, what rabies is, first of all, and what it looks like in Southern Ontario. I will give an overview of something called SAMPI. So for those of you who've worked with Python, this is a really great new tool that was developed in Patrick Layton's lab by François Viard. And it is a Python library that is basically for agent-based modeling of land diseases. And so it doesn't just have to be rabies. I mean, you could model diseases in trees if you wanted to. So it is currently being adapted to a 2-species model. Currently, it's just for 1 species. So we have used this agent-based modeling library and applied it to raccoon rabies. So I will discuss a little bit about SAMPI, but not go into too much detail. But then I will be going into detail about the actual modeling process for this project. So what it is to grow a raccoon population using this, this library, what we need to do to put these kinds of data into the agent-based modeling. So a little bit about raccoon ecology and a little bit about raccoon rabies ecology. Both are very important for this. So I'll also be going over 3 scenarios that were used in this project. So we were actually testing, as I said, how useful these control interventions in Southern Ontario have been that have been applied by the Ministry of Natural Resources. And so they wanted to know in different scenarios, you know, if we use these controls up until 2025, if we use these controls up until 2020, let's say we lose funding and now we can't control for an extra 5 years, what would that actually look like? And so we are doing our best to mimic reality. Obviously, whenever you're predicting the future, you're never entirely sure if you're predicting it right. You know, as data comes in, we're trying to modify the model. So we do have some idea, at least for what the data is starting to look like in 2022. So we look at rabies in Southern Ontario with and without vaccines to see how it spreads. And so obviously, I think you could probably predict already what it's going to look like without vaccines, but I'll show that to you. And then what does it look like once we apply those vaccine strategies? And then, of course, next steps. And there's a lot of uncertainties. And one thing that I would always encourage anyone to keep in mind, I'm probably preaching to the choir for a lot of you who've worked with ABMs, but for those who haven't, I mean, we are predicting a variety of possible realities, and we are only one of those. And so what I present today isn't necessarily exactly what's going to happen. This is just based on what we've given the model. There are certain scenarios that are going to be more likely than others. And so I just always like to remind people, like, this is not necessarily set in stone. We're running these things thousands of times. And so I'm presenting to you kind of the outcome of at least 1,000 runs and the realm of possibilities. So first of all, raccoon rabies. So rabies can be in a variety of animals, and raccoon rabies is a specific strain. It's vaccine preventable, so you can vaccinate raccoons, you can vaccinate animals against rabies, and that is what is being done currently in Southern Ontario and in other areas. If you get bitten, or if you get scratched, it doesn't necessarily spread through a bite. You can be scratched by an animal who is infected, and you can become infected that way, as long as the saliva is getting into your bloodstream. So once this symptoms actually appear in humans, the disease is almost 100% fatal. So if you get bitten, if you get scratched, you're not sure if the animal is rabid or not, it's best to seek medical attention and get that preventative vaccine treatment. Raccoon rabies is widespread along the eastern seaboard of the United States. So it is quite prevalent in the eastern United States, and it's trying really hard to get into Canada, and it has succeeded, unfortunately. The Canadian government and the US government are working collaboratively, especially along the border, to make sure that this doesn't happen. But unfortunately, things can still get through. The current outbreak that we are seeing in Southern Ontario is very likely that a raccoon was moved from probably in the New York area into Ontario. It could have been over in a truck. It could have been, you know, somehow it passed over the border and it ended up in Hamilton, Ontario. So this was very likely based on genetic studies, moved from one area to another, and then it started infecting animals in the area. For this modeling project, because the SAMPI model only works with one species, we have been modeling raccoons. So yes, I know that this can be in skunks as well, and it is in skunks as well. It can be in foxes. For now, we're just modeling raccoons, but that is one of the uncertainties in this model, is that we're not accounting for other species as well that could be affecting disease dynamics. So this is an image here. Anything that's in that kind of beigeish color is the study region that I was working with. And so that star is the first case. It was detected around December of 2015. That was the first time that they detected this disease in raccoons. Now we don't know, again, back to uncertainties, we don't know exactly when that raccoon showed up. We know that we first detected it in December of 2015, but it could have been around a little bit longer than that. there might have been some cases that went undetected. As you all know, it's always the tip of the iceberg that we actually detect. So this is the epi curve. This is at a monthly scale here, showing the number of cases that have been recorded in raccoons from December 2015 through December 2015 through September of 2021. So it looks like it's tapering off. And the reason for that is because controls were put in place. So the government didn't just sit around and wait, once they knew, and there were always controls going on in Southern Ontario, but once this case was detected, they went full throttle and really started to clamp down on this disease. And I think for the most part, they did a really, really good job using a variety of control interventions. But, and the data are not shown here because I haven't been given them yet because they haven't been finalized, but the disease is still ongoing, right? The outbreak is still ongoing. We're still seeing cases in the Niagara region. So I don't know if you can see my mouse. But towards that Ontario-New York border right there, east of that star, we are still seeing cases. So it is persisting, and we're trying to figure out why that is. So to discuss SAMPI a little bit. SAMPI was developed, as I said, by Dr. Francois Viard. This is in Patrick Layton's lab. And this is a library that right now has been modified and adapted from the Ontario rabies model, also known as ORM. So Aaron Rees and a lot of people have worked with ORM. A lot of people in Patrick Layton's lab have worked with it. And it is an agent-based model that we were using before. It's a bit slow. I'm not going to lie. It was kind of painful because it would take sometimes up to 10 days to run one simulation for what I needed at the scale and at the spatial resolution that I needed. So it wasn't terribly efficient. Now we know that it works. Obviously, it's been used a lot in the lab. It's been used for smaller spatial areas. And if you're willing to wait and use supercomputers, yes, you can definitely use it on bigger areas. But we needed something faster, and we needed something that could be more easily adapted to other diseases and adapt it for different inputs if we wanted to change things. And so SAMPI was born from that. And so, as I said, it's a Python library. It's now up on GitHub. And the paper has just been submitted. So that is out the door. We're very happy with it. And I'll show you kind of what it can do. Just to give you an idea, as I said, one of the simulations in ORM took about 10 days to do. In SAMPI, it takes about 15 minutes. So 10 days, 15 minutes. Very, very efficient, comparatively speaking. So what are agent-based models? For those of you who have not worked with it or just learning about them, they're really fun. Basically, what you're doing is you are modeling each agent independently. So in this case, an agent would be a raccoon. I'm interested in what the raccoons are doing, where they're moving, how they're dispersing over time, how many babies are they having, how is that affecting disease dynamics, how are controls being put into this. So there's a lot of information in these, and each agent acts independently of all the other agents. So if you've ever played a video game, it's kind of like that in a way. You go up to an alien and you're trying to shoot it, but then the other alien reacts to that, and they're all kind of reacting to each other and acting independently of each other. That's very much what agent-based modeling is about. And so you have to handle a lot of information in these, and that's why most of my models are run using Compute Canada, because we need supercomputers for this to run thousands of simulations. We need very, very powerful technology for that. So this also incorporates spatial data, and I'll get to that in a second. So SAMPI input factors, you have to know a lot going into this. You need to know all the information about your biological traits of your host. So in this case, we're looking at raccoons. This is very well studied. We know timing of birth pulses. We know what litter sizes tend to be like. So we use probability distributions for these. We know natural mortality rates, how long the host is going to live for. Another important thing is the home range size. So how far do these animals tend to travel? When do they disperse? At what time do they disperse? You can actually go into even more detail. In the case of raccoons, we go into detail about female versus male dispersal. Males tend to travel a bit more than females, juvenile versus adults. So there's a lot of detail that you can put into this, but that makes the model a lot more specific. So that's actually a really great thing. So my background is in GIS and remote sensing as a medical geographer. So we do a lot of mapping. For SAMPI, we had to put in a map of K values. K values are carrying capacity values. And so what this means is you have to provide the model with information about where these hosts like to be, what areas are more likely to lead to higher survival rates or lower survival rates. So in this case, we have at a 10-kilometer resolution, this is actually, you can't see from the scale, but this is made up of hexagons instead of squares. So we have hexagonal grids here where each grid cell is 10 kilometers squared cell, and it has a K value or a carrying capacity value associated with that cell. This is based on literature. So I had to go through the literature. I had to figure out where raccoon densities tend to be higher, where they tend to be lower. Obviously, they're going to be zero in areas where there's water. So you can see in all these areas here that are yellow, I don't know if you can see my mouse, those are water areas, and the rest of this just wasn't even included. And then there's little ponds and lakes and things like that. This area right here next to Lake Ontario is the GTA. You definitely do not want rabies to get in there. It will explode, as I will show you soon. So we have areas that are very suitable, unfortunately, for raccoons, which is where the rabies outbreak started. Areas that are more better suited for raccoons tend to include suburban areas. They tend to include deciduous forest. They're not crazy about boreal forest. So up north, you can see in the Algonquin Park area up north here, the values for K are a lot lower. It's because they don't do that well in boreal forest. They tend to be very well adapted to cities and suburban areas. There's a lot of trash cans and things like that. And so they've adapted quite well to that. They prefer the deciduous forest. And they also like cornfields. This is something that I learned doing this project. They like corn. I don't know. That's what they like. And of course, I live right next to a cornfield in a deciduous area, which is a suburb. So we've got a lot of raccoons around here. And so all of this gets taken into account in this map. And so this is what you put into SAMPI. So it knows kind of how to build a population based on this. And then you've got the epidemiological factors. So you need to know a lot about the disease that you're going to be studying. What is the transmission rate? What are the incubation and infection periods? Chance of death, location and timing of the first cases. Now, this last one, location and timing of first case, this is tricky because we're never sure exactly what the timing is. So there will always be uncertainty, at least with this disease, and in this particular project, about exactly when and where the first cases started. We know where it was first detected. There might have been other cases that we did not detect. And so that's just the way it works. So we've put all of this into the model. And then it gets even more interesting because we needed to know the location and timing of the control applications. So we know that the Ministry of Natural Resources and Forestry poured a lot of money and a lot of time and effort into trying to keep this outbreak from spreading further. And they were particularly worried about this disease spreading into the GTA. And so they really slammed the south of the GTA with a lot of bait stations, a lot of trap, vaccinate, release projects, things like that, that can really just try to limit the spread of it as best as they could. And so this is just an image here of a blister pack that they use. Apparently it smells like cotton candy. You know, they might be chucking it out the door of a car as they drive along in a suburb. They might be throwing them out of airplanes. They did whatever they could to vaccinate the raccoons as much as they could. And so this is, again, where GIS comes into play. So the Ministry of Natural Resources gave me GIS information about when and where these controls were applied. Again, not always very precise. We've got some areas that are kind of bubbles, which hopefully did encompass where they controlled, but we don't have that precise a data set in terms of, you know, they didn't exactly apply this every single week, you know, or at the exact same level every time. But it's a start, and it's certainly better than nothing, and it's it's enough for what we needed for the purposes of this project. And so some of these dots here that you see, those points, those are bait stations. Some of the lines that you see are helicopter paths that they took where they were dropping vaccines in rural or suburban areas. And then you've got these huge swaths, which are the airplanes, which were trying to cover broader rural spaces and just throwing vaccines out of the blister packs out of the airplanes. And again, I know what weeks they were applied, I know what years they were applied, and I know generally how much or how effective they were. So this is what it actually looks like in reality. So this is a map that was provided. It was updated around July of 2022. And it's a little bit blurry in this image for some reason. But basically, all these greens are negatives. So that's good. It doesn't appear to have yet gotten into the GTA. So pat on the back for the MNRF, because that's what they were really, really worried about. But unfortunately, it's still there. We are still finding cases ever so often. So the active cases are these purple circles here. The gray ones were cases that are now expired. And there are some Fox cases as well, but we didn't worry about that for this particular project. So that just gives you an idea of the spatial spread of the disease. And then this is what SAMPI does. So we put all that information into SAMPI and we run it. And of course, I'm oversimplifying how long this actually takes. But this is an animation that shows if you did not use control interventions, what would the spread look like? This is really fun because it actually shows you the dynamics of the disease over space and time. So this is at a yearly resolution. I'm running this from 2015 to 2022. So every time it moves, that's a different year. And it looks like donuts that are kind of radiating out, and there's spaces in between. Now, before I run these, I always try to think in my head, what do I expect to see? And I try to do that before I actually run the simulation so that I'm not as biased. And I think, okay, I thought this would just look like it would just spread. It would just be, the whole thing would be red or purple in this case. And it's not. It actually shows this kind of radiation where it's leaving a lot of death in its path. And what's happening is the disease is actually killing almost everything in its path, right? There's nothing that's limiting it. So it's spreading, it's radiating out from that Hamilton area, which is where the first cases were. And because it's killing the raccoons, there's actually nothing there to infect. And that's why the whole thing isn't a huge purple. There's actually spaces in between those waves. It's because the population of raccoons actually has to recover before another wave can go in and infect them. So here's the issue. So we actually, when I first started this, and I was, doughy-eyed, and I thought, oh, this is going to work on the first try, and I ran it. And we put in all the vaccines, and I thought, all right, this sucker is going to be completely limited by probably 2022, 2023. That's not the case. So this simulation is showing what it would look like according to the model if we applied those control interventions at the levels that they claim to be at. The level meaning every intervention has what's called a vaccine level, which is the proportion of raccoons that should be vaccinated after an 8-week period. So after 8 weeks, you go back, you sample that area, what percentage of the raccoons in that area actually got vaccinated. Things like trap, vaccinate, and release have about a 60% vaccine level. That means they're vaccinating successfully about 60% of the raccoons in the area. That's the best you can do with these controls. Then you have things like bait stations. Those are about 20%. So obviously not nearly as good as TDR. And of course, TDR is very expensive. And it requires a lot of people power. So you're not going to be able to do that across the whole landscape. So they've predicted, they being the MNRF, have predicted the levels for each of these. And so I tend to think of it as, how good are they? How many raccoons can they possibly reach? And in order to determine that, they go into the field and they sample about 8 weeks after they've applied the interventions. And they try to calculate that percentage based on titers. So they set a threshold, they take blood samples and they say, okay, if we set a threshold of so many international units per mil, that's, that raccoon, if it's above that number, it's going to be considered vaccinated. If it's below that number, it's not going to be considered vaccinated. So that level is dependent on the titer that's being used. And in Canada, we're way more conservative than in the States. So in Canada, we're using about like 50%. So if the raccoon is below that, it's counted as not vaccinated. And in the States, they're using about 12.5%. And so I can't go into too many details about that just because I would need Tor here to explain all the details about how those samples are actually measured. But what I do know is that we're far more conservative here. And I think we're being a little bit too conservative. So what we're seeing here is the disease should spread everywhere. So if we are using the levels that the MNRF thinks are the levels that are actually happening, it should have spread all over the place. And we know that it didn't. It's actually tapered off. It's only hanging on in a couple of places in Southern Ontario. This didn't happen. So probably we need to increase those vaccine levels. So that is kind of a scary image. Now you can see that because the controls are being applied, those spaces between those waves aren't really there anymore. They are a little bit near the end. You can kind of see it starting to spread outside the control zone and take off, but it's actually kind of more smushed. So you can actually see it's a little bit more pink, it's more spread out because more raccoons are vaccinated and the populations are recovering faster before getting reinfected. One thing that, a few things that we know that we were right about the transmission rate. Now, when I say right, I mean, we can't do better than we have in terms of the transmission rate. We're using 3.5%. This and a lot of the other inputs that we're using were used in the Ontario rabies model, and these have been tested and they appear to be working really well. This is just a quick example of what I did to test the Ontario rabies model against SAMPI. And so we use like a synthetic tube. We've run tons of simulations, sensitivity analyses, calibration analyses, trying to make sure that these other inputs seem to be behaving properly based on previous research. So the Ontario rabies model, we know the transmission rate, incubation period, all of that. We use a synthetic tube of cells. We run the disease and we see, is it basically doing the same thing that ORM was doing? And it is, based on the graph below. So we know the transmission rate is around 3.5% or so. And things that we don't know, which is what we had to play with here, we had to kind of experiment and do some calibration tests, were the vaccine level. So again, that vaccine level really bugs me because I don't think that those numbers are high enough. I think the Ministry of Natural Resources actually did a better job than they think they did. So I think they're underestimating just how effective they were at preventing the spread of this disease. And I didn't actually get any arguments about that when I approached them about this. Right away, they were like, yeah, they're probably too conservative. So good, that's one thing that we were able to experiment with. The other area of major uncertainty is the start week. What week do we start in? So you have to tell the model when you start these simulations, when the disease appeared. So you could put December of 2015, you could put the first week of December. It was probably around before that, right? It was detected in December, but it was probably around before that. How long before? Again, we don't know. So this is where we did calibration tests. So I ran a combination of vaccine levels that were multiplied by a certain factor. So obviously, if you multiply it by 1, you get that animation that I just showed you, and we know that that's not representative of reality. So then we start to slowly increase that number by increments of 0.2 up to about a value of 3, and then we see where we're at. And then if we see, okay, we're getting close, the epic curves that these models are producing are pretty close to the epic curve that we're seeing in reality, then we can kind of tinker, get a little bit more specific, increase those increments to 0.1 and try again. So we started with this, and we also manipulated the start weeks. So we started at week 40 through week 48. So week 48 is around the last week of November. That's probably around the time that the disease showed up, or the diseased raccoon. Why week 40 to start with? I don't know. We could have gone earlier than that. We just started with this. We saw what epi curves were produced, and then we revisited it and said, okay, this looks like this is about the right input. Let's run it again. So we ran this hundreds and hundreds of times, and we finally came up with this. So There are a few different possibilities that we could use in these models. We could use week 47 of 2015. We could use week 43. The start week didn't make that much of a difference. So from a sensitivity analysis side, that was kind of interesting. The start week could be a little bit flexible. One thing that was making the biggest difference was that vaccine level. And so if I multiplied it anywhere from 1 to 1.8, 1.9, it didn't make anything close to reality. As soon as I doubled it and took all those vaccine levels that were predicted, multiplied them by 2. So instead of thinking that bait stations vaccinate about 20% of raccoons, I'm now saying they vaccinate 40% of raccoons in the area, then we're getting a lot closer to reality. So we're predicting, as of these tests, that that level is around multiplied by a factor of 2.1. And this is actually far more realistic, I think. In previous models by Aaron Rees and others, levels needed to be around 50 or 60%. So most of these areas, like helicopters that were predicted to reach about 24% of raccoons, this brings them a little bit closer to 50, 60% in that area. So this is, to me at least, a lot more realistic than the levels that we were provided. And yeah, so far I think that's really, really good. So we started with week 47. Again, you could play with that a little bit, doesn't really matter. We did chaos tests and correlation tests to make sure that we found the epi curve that had the closest fit, and it was this one with vaccines multiplied by 2.1. So that alone already, before we even ran the final simulations, was a really interesting and important finding, is that we need to be more aware of the levels that we're using and revisit how those levels are being measured in the field. So now we're ready to run our final simulations. So we ran 1,000 simulations for 3 scenarios. So the first scenario, we ran the models from 2015 to 2025. The MNRF is predicting, hopefully by 2025, we've killed this thing off, it's gone. So we ran it until 2025. We have data up until 2022 right now. And the question for scenario 1 is, if we get funding and we can keep on controlling until 2025, what's the outcome going to look like? Scenario 2 is, let's run this until 2020. Let's imagine that from 2021 to 2025, we don't have enough funding. We don't have enough people power. We can't control it. What's that going to look like? And this is important to go back to the government and say, this is why you need to fund this project. This is really important. Look at how much further it spreads. Look at how many more animals and people are going to be affected if we don't control this. And then the third scenario is like a control scenario. So we control in the sense that we don't apply any vaccines at all. And we see how far it can spread. So this produces a lot of different outputs, right? We run this 1,000 times each for each scenario. In some scenarios, before the rabies could take off, it actually dies. So maybe the raccoon just happened to die right before it was able to spread it right at the start, in which case we have no rabies for the rest of the simulation. That's why it's important to run this at least 1,000 times to account for that variability and those different possible realities. That was a possible reality. It would have been nice if that one raccoon that came here got hit by a car or something and it never ended up spreading rabies. But unfortunately, that was not the case. So what I did was I split these graphs that I'm about to show you into 2 groups. Because it's too hard to show you, it's hard to interpret these graphs if I show you every single scenario or every single simulation across the 1,000 simulations, because it looks like they all spread. So what I did was I split it into scenarios, or simulations rather, where rabies dies off. So what does that epi curve look like? And then in simulations where rabies actually escaped and spread. So what does that look like? So this is scenario 1. So let's say we control up until, at the top rather, sorry. So in the top graph, we're looking at scenario 1. We control until 2025. For simulations where we get rid of rabies, we're pretty much done with it around 2019 or 2020. We know that that's not the case. That's not the reality that we ended up living in. So we should have been able to get rid of it using the vaccine levels that I'm working with 2.1. The bottom one is if we had controlled up until 2020. Again, they're about the same. So for simulations where we run this thing And the sample sizes are shown here. For scenario 1, where we control until 2025, about 674 out of 1,000 simulations, rabies is gone. In scenario 2, where we only control until 2020, about 603 out of 1,000 simulations, rabies is gone. So they're not too far off. They're about the same, but what's going to make a difference is the simulations that we look at left over where rabies spreads. So this is what it actually ends up looking like in those two scenarios. So the top scenario, or the top graph, is scenario 1. And you can see we control up until 2025. Now, one thing that's important to note is that these vaccines start getting scaled back around 2023, 2024. And so you're starting to see rabies can actually kind of sneak around those areas where they start scaling back. The reason the MNRF starts predict that they're going to be scaling back was they thought they'd have gotten rid of it by then. And so we're just trying to push it towards that New York border, and then the Americans could worry about it. So that was the idea. Unfortunately, the model is saying, well, if you scale it back, then it's going to escape. And this is what it's going to look like. In areas where in scenario 2, so that bottom graph, where we only vaccinate up until 2020, it's hard to tell from here because of the scale of that y-axis, but actually it takes off a lot sooner because the controls stopped in 2020. And so now the rabies actually has a lot more time to grow. So in this case, we're looking at up to 15,000 cases a month, which is insane. And so in this case, that's what it would look like between those two scenarios. Now, if we had no controls at all, 818 simulations out of 1,000 predict that rabies would take over and it would spread. And I'm about to show you maps because epi curves on their own give you some information, but they don't give you the full story. Predictably here, yeah, it takes over, and now we're at 15,000 cases at the most, and it's cyclical. And the reason you're seeing these cycles is because, you know, raccoons over time die off. You have the winter where there's a lot more die off, and then there's a birth pulse in the spring. And so you kind of have these little dips, and that's to be expected. In the situations for scenario 3 where rabies died off, it died off right at the start. So there were about 100 or so, 190, where the reason rabies had died off was because those first few raccoons that were infected never were able to spread it. Probably they just didn't the transmission rate, you know, was low, and then other raccoons died, who knows. But that's the beauty of using agent-based modeling. So we did account for that as well. So this is the map of the spread in scenario 1. So this is where the epi curves are good, but this is going to show you a lot more information to add to that. So what I did was I took all 1,000 simulations for each scenario, and I calculated the percentage of simulations where each of these cells was predicted to have at least one case in the study period. So from 2015 to 2025, how many of those simulations out of 1,000 predicted to have at least one case in each of these cells? This is important there are different ways of looking at this. This is just one of many maps that I could make and one of many animations that we could look at. This just gives me kind of an idea of where rabies would be predicted to go if it were to actually escape. So areas that are very purple, obviously, are going to be the areas where the outbreak started. Almost 100% of those are predicted to have rabies in every single simulation because that's where it started. It's far less likely to go south towards the Niagara region, but it is far more likely to go north into the GTA. Now, this is this is actually surprising, and I think the MNRF was surprised that it didn't go north, because those areas in and around the GTA are very suitable for raccoons. And so my prediction here would be this area where they really slammed it with vaccines, they did a good job. And they really prevented it from heading north. So the simulations are saying, hey, this should have gone north. And we know that it has yet to go north. It is holding on. There are 1 or 2 cases still popping up in and around the Niagara region. Simulations are suggesting that that's kind of unusual. That was probably not as many realities, possible realities, predicted that it would go that way. But a couple of them did. You can see areas where it's just a little bit beige, where it's trying to head in that direction. Now, if we look at scenario 2, it's a lot, it's spread quite a bit more. And so this is where the controls stop at 2020. And so at that point, it really spreads, and it's able to spread in a lot of different directions, including the Niagara region. And then scenario 3, where we just didn't use any controls at all, is like a death zone. So this is definitely not the situation that MRF wanted, and they prevented this from happening. So thank you so much. So in almost 100% of the simulations, rabies just spread north and south, pretty much everywhere. It starts to struggle in all of these scenarios up north here. I don't know if you can see my mouse, where it looks like little fingers are trying to reach up a little bit further. It's starting to hit that boreal zone, and the raccoons don't love it there. So it starts to slow down and taper off as it tries to spread north. So the overall finding here that I've said a few times now is those vaccine levels need to be revisited. There are a lot of uncertainties around those. It also could be that we should be treating different strategies differently. So maybe bait stations are around 20%, but maybe helicopters are doing a much better job, or vice versa. We don't know. There are, again, a lot of uncertainties around this, but it would appear that control strategy is active than the MNRF predicted. Right now, we're again predicting around 2.1. I've run this a lot of different times. 2.1 keeps showing up, or at least a factor of 2. Again, that sounds about right based on what we've seen in the United States and in other areas. Yes, so again, you also have the issue of accuracy. So detected cases that we can compare the models to, we're dependent on detection. We're dependent on how accurate those are in terms of timing. I took the earliest detection date possible. Sometimes they would say, oh, this was diagnosed in March, but they found the raccoon in February. I would take February. Obviously, I'm trying to get as precise as I possibly can. And then one thing that we cannot yet use, and we are hoping to do so in the coming months as we work on adapting SAMPI for 2-species model, is putting skunks in there. So skunks might be playing a role that we're not accounting for in these models. And so it would be nice to look at that as well. As I said, these are a variety of possible realities. So there are thousands of different possibilities. We only know as time goes on which reality we seem to be falling into. But this is really important to guide field work. So you can't necessarily say, oh yeah, this is my agent-based model. This is exactly what's going to happen. Definitely, no doubt about it. You say, well, this is a realm of possibilities. This is more likely, this scenario is more likely than this one. We can go back and revisit the data, see if we can make it any better. So thank you for coming to the presentation. I hope that you found it as fun as I did doing it. And I'm happy to take any questions that you might have.
Thank you very much, Emily, for this fascinating presentation. It was a really great overview of the topic and what modeling can do in relation to that topic in an area that's really particularly relevant to One Health. Rabies is, of course, kind of one of the birthplaces of One Health interventions. If you think about early on, how we vaccinated dogs. In the United States, for example, in the 1960s, we had 10,000 rabies cases in humans per year, as well as 10,000 rabies cases in dogs, and those were linked. And then we started vaccinating animals, and we could drive down rabies exposure dramatically in the United States. It's been below 100 annually. I think it was 2 last year. So it's really, come down completely. And the intervention was really successful. And so this is an interesting new area now where we're realizing we have to move in terms of not just the domestic animals, but also on the wildlife side, where we need to apply the One Health approach really and look at how the interventions in the wildlife sector can have impacts on human health over time. And so I want to open up the floor. We have 10 minutes roughly for questions. And there's 2 ways in which you can ask questions. Either you can pose them directly in the chat and I'll read them out to the group. Or you can also just turn on your camera and your microphone to ask a question directly on screen here with us. I'm going to start with the one that was posted in the chat, I'll just read it out. Question for the presenter: why can the library only model one species? Python is a general programming language. ABM is a general mathematical model. Why does the library limit Python, in your opinion?
Yeah, it can model 2. It can model 10. It's just that we have to test it and we have to validate it, and that takes quite a while. So what we've done so far is just worked with one species and really hammered it down, made sure that it's behaving in the way that we think best represents reality. And now we are working on a 2-species model. And eventually there will be more. Eventually, Francois is also working on vector-borne disease incorporation and things like that. So yeah, Python's definitely very flexible. It's just that we want to make sure before we put it out there that it is representing reality as best as we could.
Yeah, and of course it gets more complex the more the more variables you integrate, and the more you also these variables then interact. These different elements might also be interacting in certain ways, which makes it really difficult then to get at that reality and to model that reality.
Interaction rates especially are hard because we
exactly.
don't actually know in the field, how often skunks and raccoons interact and what the transmission will be there. So yeah, that all gets far more complicated once you bring in more species.
Okay, there's another hand up now. Amira, go ahead.
Hello, hi. Thank you for such a super interesting presentation. So I have a probably fairly ignorant question because I'm really very new to learning about modeling, but how much of the model takes into account what, let's say, New York is doing? Like, let's say if there's so many, let's say if they shut down their program, or if there's repeated introductions of raccoons coming up across the border, can it account for that, or is it accounting for that?
It's not currently, that's a great question. It is not currently accounting for introduction of new infected raccoons. It could. If we know enough information about that, and we know how often it's likely that a raccoon would be coming across and where it would be going and when, we definitely could incorporate that. For now, we don't, because we don't know. Okay, that's a great question because it's very relevant.
Okay.
We'll make it more dynamic over time, essentially. So you're not modeling time really. You're modeling time in terms of the extension, but not in terms of the introduction of new rabies into the closed system, whatever you're modeling. So that's a really interesting point.
Yeah. Not ignorant at all. Very, very good.
And of course, the other.
That was good.
Any other questions? Feel free to turn on your camera and post a question. If not, I would have a question for you in the meantime.
Okay.
I was thinking that somebody who's thought about rabies mostly from the angle of dogs, domestic animals, but also bats. I mean, in Ontario, we know that the bat population, roughly 2 to 5%, we're assuming, has rabies. So there's also the interaction with bats and the danger that comes from bats. Now, historically, raccoon rabies has not been a particularly concern. There's been one single death in the United States since 1968 related to raccoon rabies. Almost all the deaths are either related to bats, 70% of the deaths, or household domestic animals where you get it from your dog. And so why this focus on raccoon rabies? Has something changed in terms of that you're finding, more people are dying from raccoon rabies nowadays? Why not focusing more on the bat side of things, where it seems to be where more of the deaths are actually coming from?
Yeah, so again, great question. So we do have somebody on a different team who's interested in bat rabies. So that's definitely something that we will be working on in the coming, I want to say the coming months. I keep saying everything's happening in the coming months, but in the near future, we will be working on bat rabies. We were actually approached by the Ministry of Natural Resources to do this project. And that's because what they wanted to do was actually an economic analysis to see how much money they were spending on these controls and how well they were working. And they really wanted to get rid of the raccoon rabies entirely, and it's been holding on in the Niagara region. And so they kind of wanted to know why that was. And they have to be able to go back to the people funding them and say, hey, this is working and this is why it's working. And here's the quantitative proof, or at least, support for that. So yeah, we were actually approached. Patrick was approached with this project because of that. They actually want to use what we're finding here with those maps that I showed, and then present kind of an idea of how much money they've saved essentially by in terms of vet bills, in terms of human costs, all of that. And so that's why I was approached. But yes, bats are definitely very much on the radar. We just have to figure out how to model those.
Yeah, and I mean, I guess the raccoon rabies, part of the reason was probably because it was completely gone, and now it's coming back, whereas bats it never left. So it's probably harder to totally control it in bats. It might be impossible in bats to totally control it. So you control what you can control, right? So that might be one of the main reasons as well.
Yes.
There's another question here. Excellent presentation. I know your work is specific to raccoons. Do you see this modeling and bait vaccination programming being relevant for other wildlife, such as Arctic foxes in more northern regions of Canada?
Absolutely. We have somebody, Agathe Alibert, who is a PhD candidate in Patrick's lab. She is modeling Arctic foxes right now in northern regions of Canada. So it's very, very relevant. Absolutely. So that is being done. I think Olivia Tardy also just submitted something on Arctic foxes. So yes, yeah, definitely a lot of attention there as well. Those are a lot harder to model though.
Yeah.
From the complaining I've heard from Agathe, because they have these kind of random, really long dispersal distances that are hard to account for in a model.
Yeah, the more movement, the more random the movement is, the harder it is to model it.
And sometimes they can just go kilometers and kilometers. Like raccoons, for the most part, have kind of a set area that they're going to hang around in, and then they disperse a little bit, compared to Arctic foxes that can go miles and miles. Yeah. So she's trying to account for that.
Okay. Yeah. I mean, this model would work for a lot of different species and a lot of different diseases as well. So it doesn't have to be limited, in my opinion, to any disease. That's the whole point of it. That's what Francois wanted too, is that eventually it can be adapted, so you can go in and you can put in your own inputs, and you should be able to work with a lot of different species. Is it open source? Can anybody use it? Yeah. Oh, it is. Okay, that's good to know. Yeah. We should advertise it as part of the OMNI network. I wasn't aware of that. That's great. Oh no, absolutely. He wants more people to work with it, and he's open to getting suggestions as well if people have questions. Okay, that's great. That's fantastic. Do we have any other questions? Please turn your camera on, mic on, or just pose a question in the chat box. I didn't notice anything else. Oh, Jack's coming in. Hi, Jack. Go ahead.
So you did this for Ontario.
Yes.
I suspect it's easy transformable, it can be transformed into other provinces. Is the data as good in all provinces as it is in GTA and the regions? Because the concern you mentioned was mainly on Niagara, or is that Niagara region where it's observed more? Yeah, it's observed. It's still holding on in the Niagara zone, close to the border. And so we don't know why that is. It could be that just infected ones are coming through, or the disease was successfully pushed south from, instead of going to the GTA, it's kind of turn and come back this way. I haven't looked at data for other provinces because I've only been working with Ontario. I know that we do have work that's being done in the north. This is all in Patrick's lab. We have someone working on mongoose rabies in Puerto Rico. And she actually flies down there, I think. Patrick is probably on the call, so he can always fill in more detail. The data in Ontario is as good as it's better than I expected it to be, honestly. But it's not perfect by any means. The GIS data are, they could have more detail. I think it's probably hard for them to provide that. So, if you have some helicopter files, for instance, are lines and are very specific. I know exactly what cells got hit and when. Then you have fixed wing control interventions where they use fixed wing airplanes, and they gave me like a huge swath and said it was between this week and this week. And that could be better. So yeah, I don't know about other provinces. Patrick might, Patrick's there. Yeah, come on, Patrick. He might be able to give even more. I could just complete that. See, there's Alain Aspirot from Quebec is here from our Quebec Rabies group with the Ministry of Agriculture. We in Quebec, other provinces are also doing raccoon rabies surveillance, but it really is dependent on the epidemic situation. In Quebec, for instance, we recently managed to push raccoon rabies south of the border. So what we're doing there in terms of modeling is working with the United States
in the limiting areas to develop models of potential reincursion into Quebec, and then what scenarios we would need to drive to continue using barrier vaccination to drive it further and further south in collaboration with the state. So the problems are a little bit different, but I would say the data are as good in Quebec, but we didn't have as huge of an outbreak as Ontario had in terms of the absolute numbers. Yeah, and I think, and there's other, in New Brunswick, there's similar issues for raccoon rabies. And of course, there's other strains of rabies that are being monitored elsewhere in other provinces. But, Ontario is unusual in having had such, a focused large outbreak and a very active team doing, millions of tests. And I mean, really, the data set there is quite incredible when you look at the effort that was put into it.
Okay, thank you very much, Patrick.
Yeah.
This was a really lively
Patrick.
discussion. I think this is a really interesting topic, and it's one that's very relevant to One Health and really kind of, contains a lot of the elements that are really exciting about One Health modeling. And so I'm very glad we got this presentation, Emily, today. I want to give you 2 minutes to get ready for the next Zoom. Most of you will probably be in Zooms again. So I want to close this session and want to highlight that there will be another session coming up, I believe, in 2 weeks, but the information will be sent out. You already invited somebody else, and we're looking forward to as many as possible of you joining again for the next Super Spreader Seminar series of the OMNI-RÉUNIS network. Thank you very much for your